Hello class. I am to discuss a little bit about some of the principles involved in blotting techniques because I know that some of you are asking for this PowerPoint presentation and also the recorded discussion, although this is just a topic which is already understandable and procedural. Okay, so we have different blotting techniques and I will discuss only three, the most common, which are southern blot, western blot, and also the northern blot. Okay, so by the name itself, what is it that makes blotting techniques as a blotting procedure, or why is it that we named it as blotting techniques or blotting procedure? So blots are because we are transferring the nucleic acids and proteins, okay, into a different carrier, especially if we want to separate them. Okay, like for example, you have a gel medium here which contains the fragment of nucleic acids or proteins, for example, after separation, let's say after electrophoresis, and then you overlay it with another medium. Okay, so you overlay it with another medium, and of course, whatever fragments formed in these areas from, let's say, the electrophoretic gel medium to the blotting medium, okay, will be copied. Okay, so you can have again the southern blot, the northern blot, and the western blot. Okay, so I'll just have a mnemonics on this one so that you will easily remember on what type of substances these blotting techniques are used. Okay, so we have this one, snow drop. Okay, that means, okay, take note of this one, class. Southern blots, okay, are used to study what? Okay, DNA. And then, northern blots, okay, northern blots, class, okay, this one, the northern blots are used to study what? Okay, very good. So, these northern blots, okay, are used to study RNA. And of course, lastly, we have this one, W. So what is this one? Western blot. So Western blot, class, is used in studying proteins. That's why this mnemonics is often used for differentiating, okay, your southern, northern, and western blot. Okay, and again, these techniques are used in studying DNA, RNA, and proteins respectively. Okay, so this one, class. So southern DNA, northern RNA, and western blot, we have proteins. Okay, so let's have first the southern blot. So this was developed, of course, by a person named Southern in 1975, thus the name southern blot. Okay, it detects DNA again in the sample. Okay, and the DNA detected can be a single gene class or it can be part of a larger piece of DNA, such as, for example, the DNA of a virus. Remember in your virology, the viruses are classified according to the genetic material that they have, either the DNA or RNA. Okay? This one, southern blotting combines agarose gel electrophoresis. I have mentioned this earlier that from that gel we will blot, okay, the nucleic acids. In this case, in southern blot, we will blot the DNA, okay, after it has been separated according to its size. Okay, so the separation of DNA, again, with the methods to transfer the size, the fragments, okay, separated the DNA to a filter membrane for, let's say, for example, you want to perform probe hybridization. Okay, so you have this hybridization when we are talking about southern blot. So when you say hybridization, what is that one? You make a hybrid. So meaning to say, how many strands of DNA do you have? Okay, hybrid that contains double strands of DNA. Okay, double-stranded DNA molecule between single-stranded DNA probe and a single-stranded target DNA, okay, of the patient. Okay, so you have the double-stranded DNA class between the DNA, which is single-stranded, okay, found in the probe, and again, the patient DNA, which is again still single-stranded. Okay, that's hybridization. Okay? So these are the principles of the southern blotting. These are just the summaries of the procedure. So number one, the mixture of molecules is separated through different methods, and the most commonly used methods again, okay, are electrophoresis methods. Number two, the molecules are immobilized on a matrix, let's say gel matrix. Why do we have to immobilize them? Because we need to bind, okay, the molecules there. And that's also why the probe is added to the matrix to bind again the molecules. That's why before binding, it needs to be immobilized first. And, okay, those that are unbound, okay, those that are unbound class are removed. It's like you wash, okay, the setup, and then any unbound probes are then removed. Okay? That's the purpose of the probe for binding, especially the one that we immobilize, we bind it to a probe, and anything that is unbound again are removed. And the place where the probe is connected corresponds to the location of the immobilized target molecule. Okay? So that's the probe class, okay, which is again connected to these molecules that we want to bind. So I know you're familiar with this one. This is just an agarose gel, electrophoresis, okay, for migration and of course for separation. Okay, this is the setup. Okay, so these are the components, layer by layer. Okay, so you have the plastic tray. So this is the capillary plotting apparatus. We have the gel, we have the membrane, and we have filter paper, tissues, glass plate, and a weight. A weight which is of course placed on top of the setup so that it will create pressure so that the membrane class, which will adhere to the gel medium, will of course have a higher affinity to the separated nucleic acids or proteins for blotting. So in this case, since we are talking about southern blot, we are talking about DNA. Okay? So these are just the steps. So there is digestion of the DNA, and I know you are familiar with this one. Okay, with the use of restriction enzyme, because again, we need to get a single gene or like the whole viral genome, for example. It depends on what type of gene we are studying. So in this case, we are using this restriction enzyme so that we can get this fragment. Okay, and the complex mixture of fragments is subjected to gel electrophoresis to separate the fragments according to what again? According to size. And just like what you can see here, those smaller fragments migrate the fastest in electrophoresis. I hope you still remember that one. Okay, so after separation of the fragments and after migration, the restriction fragments present in the gel are denatured. When you say denatured, class, what does it mean? Okay, if they are twisted around each other, they will be separated. The two strands will be separated. And we are using alkali solutions. Okay, and this denatured DNA, in this case, because we are again talking about southern blot, is transferred into a nitrocellulose filter. So this is the gel class, this one. Okay, this will now contain the denatured DNA after using alkali solution, and it will be transferred into a filter. Okay, by what method again? By blotting. So you can use nitrocellulose filter or nylon membrane. Okay, by blotting. That's why you need to put something heavier on top of that setup. And this procedure preserves what? Okay, the distribution of the fragments. What fragments, class? We are talking about DNA fragments, creating a replica of the gel on the filter. So that's why it's blotting. So it's very simple, right? Okay, next one. The filter is incubated under hybridization. You see? Okay, my dog is very noisy. Okay, excuse me for that. Okay, so you see, class, the filter, okay, which was used for blotting, okay, like the nitrocellulose again or the nylon membrane, okay, it's the one that will be incubated, okay, under hybridization conditions. Okay, and again, in hybridization, remember we are using single-stranded probe, right? And the probe hybridizes to the complementary DNA restriction fragment. Okay, so that we can create a hybrid of DNA. Okay, next one, excess probe again is washed away, especially those that are unbound, and the probe bound to the filter paper or the filter medium is detected by, for example, an X-ray film or autoradiography, which reveals the DNA fragment to which the probe is hybridized. So that's just a simple presentation of the southern blotting. It's true for all the blotting techniques. They have the same principles, but the only difference is they are used to study different molecules. Just remember again the snowdrop. Okay, next one. This is just the summary of the procedure. So you have here the entire DNA structure, may it be coming from a single gene again or from the entire viral genome. And then you perform electrophoresis, and then you transfer or blot the DNA, okay, from gel to the membrane. Okay, it depends on the membrane that you are using. And then, of course, the bands of DNA are transferred, and then these blotted DNA fragments are incubated with a probe. And the unbound probe, of course, is removed until such time those that are only bound probes, especially radio-labeled probes, will be the one that will be analyzed, for example, on a film or let's say X-ray, and that can be detected. Okay, so this just shows you the summary of the procedure. So purify the DNA, use restricted enzymes or restriction enzymes. You separate them through electrophoresis according to what? According to size. You have to denature the DNA to unwind them in order for the preparation for blotting. And of course, you have to block with excess DNA because again, okay, we are using probe here. Okay, and then wash away the unbound probe and check in the result autoradiography. Okay? So applications class, usually southern blots are used in gene discovery, mapping, evolution, and development studies, diagnostics, and forensics. Okay, it is used for DNA fingerprinting, preparation of RFLP maps, and of course, identification of the transferred genes in transgenic individuals. Okay, so there are a lot of methods, okay, that we are using southern blotting, or we are applying southern blotting for that one. And usually the most common, aside from this one, I know you know this mapping, okay, like DNA fingerprinting, for example, especially if we are preparing this one. So this one class, the RFLP, that means restriction fragment length, okay, polymorphism, polymorphism, okay. So it's like you can see the differences among different people, okay, their DNA sequences, okay, as also recognized by the different restriction enzymes, okay. So that's RFLP, okay. Next one, northern blotting. Okay, what is this one? Okay, we are still using blotting techniques, but in this case, northern blot, snowdrop, we are detecting RNA sequences. Okay, northern blotting class was developed by James Alwyn. Okay, southern blot was developed by Southern, but this one, it was not developed by Northern. It was just named northern blot, okay, so that it will be analogous to southern blotting because we are detecting nucleic acid still. Okay, one is DNA, another is RNA. Okay, so this one's, it's still the same. So the RNA will be isolated, will be purified from several biological samples. And of course, as mentioned in the very beginning of the molecular biology, RNA is very sensitive, so it's more susceptible to degradation than DNA. So it's still the same class. You load the sample containing the RNA on the gel, and still, okay, we can use agarose gel and separate the RNA based on its size. Okay, so this will be still separated, okay, after they have migrated according to size. The gel is then blotted the same way we do it in the southern blot by a nylon membrane again or nitrocellulose filter paper, or you can use diazobenzyl oxymethyl papers. So as what you can see here, you have sandwich, okay, sandwich arrangement. Okay, you have the salt solution, we have the gel here, class. So you have that a medium here, okay, attached to the solution. In the middle, class, you have this gel, okay, and of course, you have the weights here again, paper towels until such time, okay, you can blot that one. So before that paper towel, of course, you have that filter, okay, touching the gel. The membrane is placed in a dish containing hybridization buffer with labeled probe. So it's still the same as, again, as what I have said. Thus, it will hybridize to the RNA on the blot that corresponds to the sequence of interest. Meaning to say, since we are dealing with RNA and DNA class, we are still dealing with the correct sequences of nitrogen bases. So whatever the sequences of the bases are in the gel is the one that will be blotted, okay, by that membrane, okay? And of course, in order to do that, okay, in order to perform the binding again, we are using labeled probe, okay? So labeled probe so that we can visualize it at the end of the result. Okay? It's like in your serology, like you are using labeled, let's say, labeled antibody. So what is the label? It might be an enzyme, it might be a radio label, or any label that can help in the detection of a substance or an analyte. Okay, in this case, again, we are using labeled probe for hybridization. Thus, it will hybridize again to the RNA on the blot that corresponds to the sequence of interest. And of course, there will be washing again the same way. Okay, we do it in the southern blot, okay, to remove the unbound probe. After that, the labeled probe is detected via autoradiography or via chemiluminescence reaction, especially if we are using chemicals as labels. Okay, it's again dependent on the protocol of the laboratory and the type of method that they are using or the equipment that they purchase, of course, in the laboratory. So different probes or labels can be used. In both cases, this results in the formation of a dark band on an X-ray film. Okay? Now, the expression patterns of the sequence of interest in the different samples can be compared. The same way that you have a ladder in electrophoresis. Okay? You also have that the layman's term for that, standard or basis. Okay, so to detect the patterns of sequences of the gene of interest. Okay, so that's for northern blot. So we have a standard for the study of gene expression at the level of messenger RNA transcript. So as what you can notice here, if we are talking about RNA degradation, splicing, transcript, okay, we are talking about northern blot, snowdrop class, snowdrop, often used to confirm and check transgenic or knockout mice. Okay, what's the disadvantages of northern blot? Okay, so it's again less sensitive. So meaning to say, if the RNA present in the sample is very, very low, somewhat the northern blot can result to false negative because the method is not that sensitive. Okay, detection with multiple probes is a problem, and especially if the appropriate probe or labels are not used for the appropriate sequences of the RNA. And if the RNA samples are even slightly degraded by RNAs, because again, RNA is more susceptible, right? More susceptible compared to DNA. So the quality of the data and quantitation of expression is quietly negatively affected. Okay? This one, where did you hear this one? Western blotting. This is very popular because we are using this for the detection of HIV, right? Human immunodeficiency virus. Okay, so what is again the use of Western blot? Okay, it's the detection for what? Proteins. You have here proteins. Okay. Western blotting is an immunoblotting technique which rely on the specificity of binding between a protein of interest and the probe, still probe. The three methods use probe. Antibody raised against that particular protein. Okay? And to allow the detection again of the protein of interest in a mixture of any other molecules. I don't know if you still remember the structures of the HIV virus, right? So, the HIV virus, just a review, okay, is a spherical virus class, a spherical. Okay, inside of which class, we have this structure, virology class, I hope you still recall this one. What is this structure? The small circles class, okay, that's again capsomers. One, one circle is a capsomer, different and many circles. We have capsomers, and the entire structure class, we call it as capsid, right? And then inside of which we have the RNA, because HIV is an RNA virus. Aside from that, the HIV class has this knob-like structure and transmembrane protein structure, right? This is the GP41. Okay, the GP41. This is GP120. Okay, so you have 72 of which knob-like structure in the HIV. Aside from that, again, we have three enzymes inside the HIV. So that is reverse transcriptase, number 1, number 2, integrase, and number 3, protease. So what I will be emphasizing here, because we are talking about Western blotting, is that in Western blotting, we are not detecting, okay, we are not detecting the RNA of the virus. What we are detecting, class, are the proteins, this one, the glycoproteins, the transmembrane proteins, or the surface proteins of the virus. Okay? So let's say you have the GP120 and then GP41 and the P24. So GP120, GP41, and P24. Normally, these are the proteins that are detected in the Western blot. And if a person, okay, a person's specimen is tested, and then it contains two proteins, then that person is considered as having HIV. Okay? 2 out of 3. But there are instances wherein 4 glycoproteins are tested. So 3 out of 4 will be considered positive. So I know you have that in your serology, but what I am emphasizing here for you to understand the principle of the Western blot is that it detects the proteins of the organism or the infectious particle that you are after for. Okay, in this case, we are talking about the HIV, and we are detecting its surface glycoprotein, its transmembrane glycoprotein, and other proteins involved for the replication of the virus. Okay? So this one, class, for Western blotting, we have a protein sample. Again, snowdrop. Protein sample is subjected to electrophoresis on an SDS polyacrylamide gel. Okay? Sodium dodecyl sulfonate. Remember, the SDS will be the one to denature the protein. So if you have this protein class, okay, the SDS will unfold that one. So it will unfold the protein. And of course, when you run the specimen in the electrophoretic medium, okay, you can separate the proteins according to its size. Okay, so southern, northern, and western blot will separate the nucleic acids and the proteins based on their size. And of course, electroblotting will be performed, and it will transfer the separated proteins from the gel to the surface of a nitrocellulose membrane again. So you have here the gel, and of course, this will copy, okay, the protein, okay, the proteins present, okay, in the sample. So that's the membrane sheet. And then this one, class, the blot is incubated again with what? Okay, with a generic protein such as milk proteins, or the BSA, which binds to any remaining sticky places on the nitrocellulose. Okay, so incubation with other proteins. Okay, and what is added? Okay, an antibody that is specific for the protein of interest is added to the nitrocellulose sheet and reacts with the antigen. The one that I am talking about, for example, is the GP120 again, GP41, P24. These are protein antigens. Okay, antigens. This is a shortcut of antigen. These are protein antigens. So aside from um, incubating with proteins to really select, okay, the antigen of interest, we are using antibody because antibodies, again, class, are specific. So among the three, for example, the red, what is the color of this one? It's like blue and green. So meaning to say, this one are also proteins. The three of them are proteins, let's say, that we can find in the HIV. But if we are only, let's say, looking for GP120, we will be using antibody that is specific to GP120, and that antibody will bind to the antigen, the GP120, present, okay, in the gel, and that can be blotted or transferred to another membrane. So what is the confirmation here is that the specificity with the use of the antibody is the one that helps us in detecting specifically the antigens, the protein antigens, okay, from a particular virus or organism. Okay? After washing for removal of non-specifically bound antibody, okay, second antibody is added. Okay, and that antibody is what? The one that recognizes the Fc domain of the primary antibody. So meaning to say, the second antibody is added. For example, class ha, okay, we have here the gel, right? So we have the gel. And then we have, let's say, three proteins. Let's say this is P24, this is GP120, this is GP41. Okay? So, for example, our technique will only detect, let's say, GP120 again. So, we will be using antibody. We will be using an antibody that is specific to what? To GP120, right? So, meaning to say, it will not be bound to P24 and GP41 because, again, antibodies are very specific in their action. So, it will bind only to the antigen. Okay, this one, in this case, GP120 is the antigen. The antibody will be bound to it. And of course, we will wash in order to remove the antibodies which are not bound, for example, here. So to wash them, okay, to remove them, okay, we will perform washing. And a second antibody is used, which is directed against the Fc portion. So that antibody will bind to the Fc portion of the first antibody. And take note, we are still using labels. So in this case, the second antibody class is labeled with a radio label. So the second antibody is radioactively labeled or is covalently linked to a reporter enzyme. So it can be a radio label or it can be an enzyme label, just like what I have mentioned earlier. And that purpose class, again, the labels again and again are just used for the visualization of the antigen antibody complex. The antigen is the GP120, the antibody is the antibody specific to the GP120, and the another antibody which is labeled with a radio label or enzyme is the one that will help in the visualization. That's why we can visualize the bands after electrophoresis. Okay? So again, it's used for HIV, okay, or for BSE, bovine spongiform encephalitis. Like, I don't know if you have heard about Creutzfeldt-Jakob disease, or let's say mad cow disease. Mad cow disease, what else? Creutzfeldt-Jakob or Jakob disease. So they are spongiform encephalitis. Or what else? Scrapie, this one. Scrapie class is a disease that we harbor from sheep. Okay? This one, of course, from cows. And this one, usually, Creutzfeldt-Jakob disease class is developed by the people who are given growth hormones as their therapy. So, all of these conditions mentioned are a type of spongiform encephalitis or spongiform encephalopathy. Okay? Some forms of Lyme disease can be also tested, okay, using the Western blot. Okay? So, any question, class? Okay, so that's all for the Southern blotting, Northern blotting, and Western blotting. Very easy, right? Okay, so I hope that you understand the lesson. And please, for your exam, you study restriction enzymes. Okay, you study also the plasmids in particular. And also, of course, this one. Okay, God bless, class. Bye.